All right, let's continue. So the biochemical reaction normally can occur quickly in the cell due to the presence of enzyme that help to speed up the reaction. So like catalyst, it will help to speed up the reaction. However, remember, before and after the reaction, nothing changed on enzyme. Same goes to catalyst. It will remain the same. Okay, it just helps to speed up the process. So the substance, like there is an active site there. The active site is only specialized for that particular substance. Then when they fit in, and then they will combine into a product. So the breakup one is the body lah. When this one comes in the active site that has a breakup one, put inside, and then they will be breaking out into two. Like that. Okay, so an enzyme is an organic catalyst. Okay, that is mostly made out of protein and is produced by living cell organism. So, but not all of them are coming from protein. Remember, protein can be used to produce. Some of the enzyme need protein to build up. Maybe the content include protein, but not all enzyme is made from protein. Okay, so the substance that needed for an enzyme reaction, because substrate means that the product is called substrate. So the substrate will bind with the enzyme at a specific site. We call active site, and form and this one is called enzyme substrate complex. Okay, this and this together become enzyme substrate complex. They haven't break up yet. They haven't break up yet. Okay, so this is called the enzyme substrate complex. Now, for the naming of enzyme, okay, normally the enzyme names are usually end in ASE. For example, the substrate name, type of reaction, then ASE. For example, the substrate is lipid, so we call lipase. Urea, urease. Maltose, maltase. Cellulose, cellulase. Lactose, lactase. Sucrose, sucrase. You see, it's the front part of the substrate, ASE at the back. However, not all of them are following by this, because there's an old method one. There's a one that use old method, which called pepsin, trypsin, and rennin. These three are the what you call enzyme for protein. Okay, pepsin, trypsin, and rennin. Why? Because these three already occur. The name already give to them before the IUPMB. This is the under IUPMB standard, because this one comes first, then IUPMB comes in. So in order to so-called, normally we will say to, how to say, we will like hallmark lah for the old people one. So that's why they remain the old name. Okay. Yeah. All right. Now, for example, okay, the lactose, and water, and then with lactase enzyme, you were able to create glucose and galactose through hydrolysis, because there's a water. Means that there's the process, the what you call the reactant, or we call the substrate, need water. However, there are some of the enzymes that do not follow. Like I said just now, the pepsin, and rennin. They are not following the what you call the naming system. Okay, so let's see the general characteristic of enzyme. First, it does not act rapidly. Enzyme act rapidly, means that it's a continuous action. Done, next one comes in. Done, next one comes in. Okay, then enzymes are only required in small quantity and they can reusable. So that's why it can act rapidly. So the structure of enzyme remained unchanged and are not destroyed after a reaction. And then the enzyme are biological catalysts that speed up the biochemical reaction. And the reaction of some enzymes are specific, means that they only react on certain that specific nutrients only. Okay, it's like sucrase only for sucrose, cellulase only for cellulose. Okay, so only substrate that fit the form completely with an enzyme in active site can combine. So some enzyme need some cofactor, like for example, vitamin B, magnesium. It's like how to say, cofactor means that they need this to occur. Means that it's like the cofactor doesn't involve in the process, but they need this to occur. Like a trigger. Means that they need it's like a kunci to open up the process. Okay? So the enzyme activity can be slowed down, okay, or stopped by enzyme inhibitor. Means that enzyme inhibitor is the enzyme stopper, the substance that can stop enzyme. Okay, an example like heavy metal, like lead and mercury. That's why when you accidentally take in heavy metal, okay, you will die easily because we all of our process need enzyme. Heavy metal destroy enzyme. So no more enzyme, no more daily activities. Then eventually you will die. And most of the reaction that catalyzed by enzyme are reversible. Most of them can be like breakdown, combine. Both can be done. But not all of them. Some of them only can destroy. Some of them only can build up. Okay? But most of it, the process are able to, is reversible. Okay? So any questions up to now? Now let's see intracellular and extracellular. Intracellular are those enzymes that build out for the own cell use. They use it by their own. Extracellular are those that will be released to outside. For example, the enzymes that digestion enzyme, mostly like in your stomach, okay, your amylase and your ileal, and then your what you call that, your intestines, okay, ususa. All these enzymes are called extracellular because they are releasing out. Intracellular is the one that used within the cell. Okay, so the enzyme that are released in the cell for its own use, we call intracellular enzyme. For example, hexokinase. Okay, normally they use for glycolysis during the cellular respiration. So there are some enzymes that are secreted outside the cell, and we call this as extracellular enzyme. For example, trypsin, which produced by the pancreatic cell, okay, will secrete into the duodenum to break down the polypeptide. Okay, so now let's see the productions of extracellular enzyme. Okay, like this one. You see, there will be produced. You see, the Golgi apparatus and endometrial, what you call that, the rough endoplasmic reticulum, or the smooth endoplasmic reticulum, they are involved. Why? Because these three are the one that helps to create and modify the protein. So there will be a ribosome. Okay, ribosome is the site of protein synthesis. So the protein synthesis by ribosome enter the lumen of the rough endoplasmic reticulum and start transport through it. And then ribosome are those, this one, the small site here. Okay, so once they reach the end of the rough endoplasmic reticulum, then the membrane will butt off to form the transport vesicle like this. Okay, there are a lot of BG here. Then they will release. Okay, and then the transport vesicle, which contain the protein, will move toward the Golgi apparatus and then fuse with it. So it will like going out and then fuse with this. And in Golgi apparatus, the protein are modified into enzyme and secreted in the secretory vesicle, which are formed from the tip of Golgi apparatus. So like this. From here, it will be released out like this. See? The small one is ribosome. Big one is the, we call the secretory vesicles. Okay? And then these secretory vesicles will move towards the plasma membrane and fuse it with it to secrete the extracellular enzyme. So this one later on will go to here, and then it fuses with the plasma membrane. The enzyme inside will be released out. The vesicle now will become plasma membrane. Okay? All right? Can, so it's something like this. Why this one start here? Because you see at the ribosome here, then it enter here. Why? Because the nucleus contain RNA. RNA contain the template of the enzyme, the structure of enzyme. So they need to be near so that when within the rough endoplasmic reticulum, it can be modified. Okay? Can I think this one you need to jot down. Make it your like something like a note. Not really note, it's just for you to have a better memory. Okay. All right. So let's see the mechanism of enzyme reaction. We call it as a lock and key. Okay. So most enzymes are complex protein that make up of polypeptide chain, which are folded into three-dimensional structure. Okay, and this structure, this three-dimensional structure, will have an active site. Okay, with a specific configuration that complements a specific substrate molecule. Means that, see, each of them only comply with that particular substrate or product only, because it can be reverse, so means that the product come in can combine into substrate. Substrate comes in can break down into product. So it only comply with that particular substrate and products only. Okay, now because the binding of the molecule substrate on the active site is like a lock and key combination. So we said the enzyme is the lock and the substrate is the key. Okay, why the substrate is the key? Because the lock can be opened on and off, but the substrate is the one that comes in. You want to break down or break in to combine. So that's why enzyme is like a lock, because it can be opened up and closed. So other one will be the key. Okay, this must be remember. You must remember clearly which is lock, which is key. All right. So most of the reaction here require high activation energy. For example, the blue color one is without enzyme. You see, why is slow? It doesn't mean that it cannot be breakdown. It means that the process cannot continue without enzyme. But with enzyme, you are able to carry down, lower down the activation energy. So to make the process faster. When the activation energy means that the energy that require for the process to occur. Like for example, I want to break down needs the energy to pull and destroy the bond. So this require the energy, and that is called the activation energy here. So with enzyme, it will lower down the activation energy, makes the process easier. Okay, so this is how. So the activation energy is the energy needed to break the bond in substrate molecule before the reaction can occur. And enzyme function by lowering the activation energy. With this,